Assalamu alaikum. So for our course Practical Techniques in Biology, today we are going to discuss the ecological techniques. Consequently, aims or learning objectives for today's lecture are: why do we study ecosystems? Which techniques are used to study the ecosystems? And what is the importance of sampling techniques and random sampling? So, coming to our first aim, why do we need to study ecosystems? So if we roughly define ecosystems, ecosystems are the biological communities living in a particular area and their interaction with the abiotic components of that environment. So the interaction between the biotic components means the living components and the non-living components, they constitute a ecosystem. Okay. So why do we need to study the ecosystem? Because it allows us to compare between two different ecosystems. It allows us to monitor, model, and evaluate a particular ecosystem over time, and it can help us to find out the changes in a particular ecosystem which are due to the natural reasons or which are due to the human activities. A very good example is the greenhouse effect or the global warming. Okay, the global warming is a phenomena that has happened because of successive release of carbon dioxide into the environment. Okay, now carbon dioxide is naturally released into the environment by the living thing activities. Okay, by the respiration. But in addition to the respiration, fossil fuels burning, our vehicles, factories, they're also emitting carbon dioxide. So there is a natural carbon dioxide emission and there is a carbon dioxide emission because of human activities. So collectively, the study of ecosystem allows us to see the effect of natural conditions on an ecosystem and the effects of human activities on an ecosystem. Okay, now, so to study ecosystems, what do we need to study? We need to study the components of an ecosystem, and most of the components are biotic components. So the techniques which we are using to study an ecosystem are collectively termed as ecological techniques. These techniques aim to investigate the biotic as well as abiotic components of an ecosystem and their interactions. Field ecologists, they investigate the living communities by using different kinds of sampling techniques. These sampling techniques are selected on the basis of their capability of eliminating bias. A bias is a deviation from a particular result because of our personal interest or because of our personal opinion. Secondly, the sampling techniques are used to reduce the effect of a chance. Chance is something which is unusual. For example, a particular species A is not naturally present in an ecosystem but has been brought by an individual. So this gives rise to chance. Okay. Now, and the third thing, why the sampling techniques are used. So sampling techniques are used to minimize the errors in collection of data or making measurements or collecting the samples. So which kind of techniques are used? Obviously, we are using some kind of instruments, some kind of tools, and quadrats and transects are basically the tools which are used by the field ecologists. Now, how they are used before going to the use of quadrats and transects, we'll just have a look at different kinds of sampling techniques. So there are three main types of sampling techniques: the random sampling, the systematic sampling, and the stratified sampling. The random sampling is usually carried out when the area under study is fairly uniform and it's very large and there is limited time available. For example, we have an area of 100 square kilometers. So see, if we go on counting one by one a species of plant or an animal, if we go on counting each and every individual, so we will take ages to count them all. So what do? We take random samples, and these random samples, then we collect an average, and then we record our data. Now, systematic sampling. Systematic sampling includes a line transect or a belt transect method, and here the samples are taken from at a fixed interval, usually along a line. Stratified sampling. Stratified sampling, what do we mean by that? Strata mean like a group or area. Okay, so it is used to take into account different areas or stratas which are identified within the main body of a habitat. These stratas are sampled separately from the main part of the habitat. Okay, so let's see what are quadrates and transects and how they are used in sampling. So quadrates. So quadrates are actually square shaped metal or wooden frames of known area, and their areas can be like 0.25 meter square, 0.5 meter square, or 1 meter square, depending on the area where we are going to do the sampling. So various approaches are used to determine representative species within an area, and these particular terms are used when we are collecting the data regarding a particular species, like species population density, the percentage cover, and the frequency of occurrence. Now, species population density is number of individuals per square meter and is determined by counting all individuals within a series of randomly placed quadrats, and then we calculate the mean. Percentage cover is determined subjectively by assessing the extent to which different species occupy the subdivided quadrate frames and by use of point quadrate frames. Now, frequency of occurrence of a species is determined using subdivided quadrates and point quadrate frames. Now, here I have mentioned the method by which we are going to calculate these things. Now, let us just go one. So here we see a quadrate frame. A quadrate frame, as I told you, it's a square quadrate frame made up of wood or metal, and it is used to investigate the distribution of species. Okay, now the square quadrate without the subdivision is used to find out the population densities, while a square subdivided quadrate is used to find out the percentage cover of a species. So here you see this frame is divided by strings, and here we can see that there is a species A present in this area and species B present in this area. So it helps us to find out the percentage cover of a particular species in a given area. Let's just see how will we calculate the population density. Here we are showing an example of square quadrate of 10 cm by 10 cm. Now first of all, when we are calculating population density, what do we have to do? We have to determine the size of a quadrate in meter square. So 10 centimeter into 10 centimeter, so the total area would be 100 centimeter square, and if we convert it into meter, it is 0.01 meter square. Now after that, we will count all the individuals which are present within the boundaries of this square quadrate. So let's just count. So after counting, we found out that there are 22 individuals which are present inside the boundaries of the quadrant. Then, putting a formula, So population density is equal to number of individuals per square meter. So number of individuals 22 and the area 0.01. So the total flowers or the total population density of this particular flower is 2200 flowers per square meter. Okay. Now let's just use a subdivided quadrate frame. This is a subdivided quadrate frame and this is used to study the percent frequency of a particular species. Now, how do we calculate that? Count the number of squares with flowers. So first of all, we will count the number of squares here. So after counting the number of squares having the flowers in this particular quadrant, these are 15. Okay, so one square has two flowers. So we are just counting here the squares. Then count the total number of squares. So the total number of squares here are 36. Now let us just calculate the percentage. So percentage in this case 15 squares with the flowers divided by the total square multiplied by 100. So 42%. So the percentage cover or the percent frequency of this particular flower species is 42% in this particular quadrant. A third type of quadrate is point quadrate, and this point quadrate is used to calculate the percentage cover of a particular species. Now point quadrate is actually like this. So there are metal pins which are dropped through different points or holes, and the hits, where do they hit? Which species are they hitting? So the hits are recorded. The pins are placed through holes in this horizontal bar and there are wing nuts to secure it at a particular area, and here you see these are the vertical legs of this quadrate, and if you look at this area from top, so it looks like this. It has holes from which we drop the metal pins. Okay, now the number of hits divided by total number of pin multiplied by 100 will give us percentage cover of a particular species. Okay, so the number of times a metal pin hits a particular plant will give you the percentage cover of that plant in a particular area. Now the second types of instruments you can say which the field ecologists use are transects. So what are transects? Transect is a line created with the string or a tape along which systematic sampling is performed. So transects are particularly useful for sampling area where there is a transition of species from one habitat to another as the environmental conditions change. For example, if we are moving up the hill, okay, so the species down the hill would be different, and as we go higher and higher, the species will change, okay, because the climate is changing. Transect studies are used to investigate gradients such as zonations on rocky shores and changes in the species diversity across sand dunes. So there are different types of transects: line transects and belt transects. A line transect is the one in which all individual organisms touching the tape or string, the line, are recorded. Then belt transects. So belt transects involves laying a tape through the area of study and sampling the population with quadrats positioned at regular intervals alongside the tape. So let's just see line transects. So here you can see a deep sea diver has put a line and it is randomly placed across the area. Okay. So what they are going to do, they will just record all the species which are touching this line. Okay, here you can see another example. Okay, all species touching the line are recorded along the whole length of the line or at specific points along the line. If the line is fairly long, so they will record at specific places along the line. Then they will measure presence or absence of a species. On the other hand, belt transects are laid out and then quadrates are placed across these belt transects at specific intervals, and then samples are identified and abundance is estimated. So slow moving animals like limpets and snails are collected, identified, and then released. And for plants, a percentage cover is estimated. Now this data collection should be completed by one individual, as estimates can vary person to person. So there is a potential of having a bias in the results based on the person's approach towards taking the sample. Now here we are giving you an example of a belt transect survey of a dune system. I have taken it from a resource which I have listed in the reference. So you see here, this is a particular sand dune. So tape is laid across this sand dune. You can see here there are some plants over here, there are a few plants over here, and then there are different types of plants on top. Okay, so this arrow is indicating the distance inland from high watermark area in meters. Now, in this particular example, a belt transect was used to investigate the distribution of 3 species of grass commonly found on sand dunes. The transect line stretched from the high water mark to the inland area, and 1 meter into 1 meter quadrates were used to determine the number of individual plants of each grass species along the profile. What data we got? So here we have distance inland from high water mark and the number of plants per meter scale. Now here you see 0 to 5, then 5 to 10 meters, 10 to 15, and so on and so forth. Now if you look at the plant species A, the plant species A is found at a distance from 0 to 20 meter from the high water mark. And if you see, more number of this species are present at a distance of 10 to 15 meters. On the other hand, plant species B is missing in this area. Okay, and it starts to appear 15 to 20 meters after the high water mark. And the most of the population of this species is present at a distance of 25 to 30 meters from the high water mark. And if you see, this species starts from 15 meters and it goes till 45 meters. Then plant species C, it starts to appear 40 to 45 meters away from the high water mark, and the highest number of this species is at 60 meters away from the high water mark. This tells us that this species is not present in this whole area, and as we go away from the high water mark, the number of the plants for species C are increasing. Okay, now what you have to do is you have to present this result as a bar chart, and in our next lecture we are going to look at other kinds of ecological techniques to study various animals, birds, reptiles, and other biotic components of the ecosystem. Okay, so take care, and here are some references. You can search these websites or you can read these papers to get more information. Thank you.